NIH clinical trial of universal flu vaccine candidate begins. Vaccine targets 6 flu strains. Enrollment in a Phase 1 trial of a new investigational universal influenza vaccine candidate has begun at the National Institutes of Health's Clinical Center in Bethesda, Maryland. The experimental vaccine will be tested for safety and its capacity to trigger an immune response in the study, which is funded by the National Institute of Allergy and Infectious Diseases, NIAID, a division of the National Institutes of Health. Certain influenza strains can be prevented using the seasonal influenza, or flu, vaccinations that are now available. Each year, the vaccines are re-evaluated and changed to best match the strains of flu predicted to be the most dominant in the upcoming flu season. Most seasonal flu vaccines are designed to train the immune system to defend against 3 or 4 different common strains of flu, but a universal influenza vaccine might someday provide protection against many more. An ideal universal influenza vaccine could be taken less frequently than once a year and protect against multiple strains of influenza virus. With each new universal influenza vaccine candidate in clinical trial, we take another step closer to that goal, said Acting NIAID Director Hugh Auchincloss, M. Researchers at NIAID's Vaccine Research Center created Flu-MOS V2, a potential vaccination candidate, VRC. It is an adaptation of an earlier universal flu vaccine candidate, Flu-MOS V1, which began first in human testing in 2021 and is still undergoing trials. Flu-MOS V2 is designed to induce antibodies against many different influenza virus strains by displaying part of the influenza virus hemagglutinin, HA, protein in repeating patterns on self-assembling nanoparticle scaffolds. Exposure to these harmless fragments of virus proteins prepares the immune system to recognize and fight the actual virus. When tested in animals, the experimental vaccine resulted in robust antibody responses. While the Flu-MOS V1 vaccine candidate displays HA from 4 strains of influenza virus, Flu-MOS V2 displays HA from 4 influenza A viruses and 2 influenza B viruses. The researchers anticipate that this will further broaden vaccine recipients' immunity, providing protection against a wider variety of influenza viruses. The new clinical trial is expected to enroll 24 healthy volunteers, aged 18 to 50 years, who will receive 2 intramuscular injections of the Flu-MOS V2 vaccine candidate. At first, participants will be enrolled in the lower dose group, 60 mcg per vaccination. If no safety concerns are identified after at least 3 participants have received this dose, enrollment will begin in the higher dose, 180 mcg per vaccination, group. Blood samples will be taken during study visits to measure any immune responses to the vaccine candidate. NIH is the primary federal agency conducting and supporting basic, clinical, and translational medical research, and is investigating the causes, treatments, and cures for both common and rare diseases.